Hello, I am Ben Clutter, and I am here to talk to you today about vitiligo, current research, and therapies. Vitiligo is an acquired skin disorder, and the main trait of this skin disorder is the lesions that we see along the knees here in this patient. And so these lesions, what's going on is the melanocytes are being destroyed, and there's a couple of different theories about how this is happening. It's frequently with other autoimmune disorders, and typically that's the pervading theory that we're looking at. About 0.5 to 1% of the worldwide population is affected. Gender is not a factor, although some of the research I found did say that some types of vitiligo are a little bit more gender biased, and also uh, worldwide population, some of them it's about like 2%, where some of the estimates from 0.5 to 2%. But most research says somewhere between 0.5 and 1% overall. The differences in the vitiligo is basically how it's distributed across the body. Is it large portions of the body? Is it kind of spread around a little bit? Or is it a bunch of tiny little, almost like reverse freckles, where the melanocytes are being broken down in little spots along portions of the body? Um, it's frequently associated with other autoimmune diseases, as I had mentioned, and that's kind of one of the leading thoughts, is that this is some tie in some way to an autoimmune disease. It Again, it has a wide spectrum of impact, and so it's basically thought to have a multifactorial etiology. Uh, that means, in other words, it probably is not just any one thing that's causing the disorder. It's usually a combination of a few things. For example, it's partially immune, partially genetic, and as you may know, our immune system has a very large genetic component to it. We do inherit a lot of our immunity from our parents, and so it kind of goes hand in hand. But on the other side of the whole story is that, yes, there definitely is a genetic component, there's definitely an immune component, and there's maybe even some other things going on there. Another leading theory is that oxidative stress might be causing it, and that might just be one of the triggers for the immune system or for the genetic factors that are kind of going into the whole thing. So, again, there are multiple factors impacting the onset of this disorder. The most strong theories point to an autoimmune disease. It is frequently occurs with other autoimmune diseases, and this means that there are usually self-reactive T cells of some sort in the blood. There's a lot of times antibodies in the blood that are attacking self-antigens. A lot of time there are some cytokines being released, and again, these cytokines are the chemical communicators of the immune system, and these cytokines a lot of times are telling us, hey, these are signature traits that we see in an autoimmune disorder. So because of the similarities to a lot of other autoimmune diseases, that's the prevailing theory. You can see here there are autoantibodies that are frequently found, and those autoantibodies are frequently focusing on the pigment cells. Current theories state that the immune system may target melanocyte stem cells or melanoblasts. And this was an article by Attili and Attili. I reference it at the end of the slideshow. I didn't focus too much on that paper. I was looking at it, was thinking this would be a good paper to incorporate into a slideshow, but really that was the conclusion, is that basically melanocytes, stem cells are growing and they're trying to replace the defective um, the effective melanin cells, and as they're growing, if it's from the stem cells, and there's some defect there, then the immune system is going to attack those, and that's how you kind of get the different patterns of Vitiligo. Basically, where along the lines is the antibodies attacking the stem cell line. If it's attacking more along the melanocyte stem cells, then yes, that's going to basically cause a much larger area of disruption. It's attacking more of the root of the problem. If it's the melanoblasts, it might be a little bit more concentrated or in smaller areas. And so one idea is to try to figure out how to target the different pathways. And it's basically this thing where there's this constant battle going on between the immune system and the melanocyte stem cells. And it's just thrown out of homeostasis, and that's why you end up with the disease. But that aside, there wasn't much more to that article, so that's why I did not include that one in this presentation. Some hypotheses are that there are activated cytotoxic T cells or CD8 cells that are attacking the melanocytes. Others are that there are antibodies. Some of the associated inflammatory cytokines that I mentioned are TNF alpha, interferon gamma, and then some of the interleukins 10, 1 beta, 6, and 8. Particularly, I'm going to take a look at one article on interleukin 8 in this presentation. So Farag and others in their article, they studied the role of interleukin 8 on this whole process. It may have value in determining if this is the cause of the Vitiligo. It could also take a look at whether this is somewhere in the process, like some way of detecting it, maybe an early detection type of method, and also just kind of give us a better idea of what's going on, why these melanocytes are being killed off by the immune system. So IL-8 is a pro-inflammatory cytokine. It's associated with some other skin disorders such as psoriasis. Some of the articles I was looking at basically talked about whether there was itching or not with the vitiligo. That's a common characteristic. It's more common and more severe in people with vitiligo, but it seemed to me like it wasn't like a defining characteristic. It could be because of the IL-8 interacting with the neurons, but it also may have just be a result of the immune response. It's generated by many different cells, such as neutrophils, monocytes, some of the endothelial cells, as well as keratinocytes. And it's going to interact with a cytokine receptor 1 and 2. They're found on monocytes, neutrophils, CD8 cells, which I had previously referenced on a slide. And these are the cytotoxic T cells. And so what some scientists believe is happening here is that the interleukin 8 is drawing these CD8 plus cells in and causing them to attach to melanocytes and then ultimately kill them. Natural killer cells, myeloid-derived suppressor cells, keratinocytes and melanocytes, both are going to have these receptors. And so what this will do is it will initiate the migration of the neutrophils, and then the neutrophils will then go through adhesion to the endothelial cells. They'll enter into the skin, and then it also will activate these neutrophils and have them start to attack the melanocytes. It may also attract T cells to vitiligo lesions. It also enhances the inflammatory reaction and melanocyte destruction. And then it's also linked to the apoptosis of keratinocytes. And so when they get destroyed, they release more inflammatory compounds, also particularly more interleukin 8. And so it's kind of this downward cycle where the more interleukin 8 you end up with, the more attraction of the immune cells to an immune response, and then you end up with more interleukin 8, and now you have more widespread vitiligo. So what we're seeing here is this is serum IL-8, so interleukin 8, this is the levels that were measured. Here we have IL-8 mRNA, and so mRNA is what tells us to transcribe interleukin 8. And so the scientists took a look at what these levels were and compared them to a control group, which were non-vitiligo. So here's our vitiligo group, 26.25, and then the mRNA was at 8.48. And when we compare that to the control groups, it was 0.57 and 0.60 respectively. And so basically that gives us a very statistical probable analysis that yes, there's definitely something different going on. Another thing that they noted was the IL-8 serum levels were different from male versus female. Notice the male is much, much higher, and that's because the IL-8 expression has some tie to testosterone levels. And down here we can see the mRNA levels in both groups. So here are the serum levels that we see in the vitiligo group, and here we have the ones in that were non-vitiligo, the control group, and same thing here with the mRNA transcription levels. So there was increased serum levels and mRNA expression in the vitiligo patients. And so this basically means that IL-8 probably has an active role in the vitiligo development. And so this could possibly be a therapeutic target if we could just get that downward spiral of IL-8 production under control. It might help alleviate the immune response. I would think that it probably wouldn't be an end-all. It would be more of just a therapy to kind of keep it under control. In another study by Lee and others, they looked at what initially I thought was going to be an immune article, but it was more about genetics, because as I had mentioned previously, many of the components of our immune system are inherited from our parents, and one thing that scientists have to look at quite frequently is HLA, or human leukocyte antigens. Particularly this article looked at human leukocyte antigen A. So disease has been linked to a hereditary influence, and so what happens here is MHC1, which is one of the versions of HLA, is a major histocompatibility complex, class 1, and it's going to be responsible for the display of non-self antigens to CD8 or cytotoxic T cells. And I got cut off here in the video. You can't see it behind this picture, and that was because of the importing of the slideshow, and it shuffled around. But I will include the actual original slideshow. But sometimes that happens, and I wasn't going to re-record the previous portions of this. So yeah, so what happens here is this is MHC1 over here. And so this displays particularly to CD8 or cytotoxic T cells. And so when this attaches, it attaches to the to the cytotoxic T cell. It releases these perforins, and then that ends up killing the cell. So here it's being displayed. Here's your cytotoxic T cell. It will recognize that and then make it release these perforins that go down and then eventually kill this particular cell. And so that could happen with a melanocyte. And so what kind of the gist of it is that we inherit our HLAs from our parents, and particularly HLA has some susceptibility to being more likely to attach to the melanocytes. Basically makes it more likely to be self-reactive and accidentally recognize self-antigens rather than non-self-antigens, because of the variety of antigens that we have on our immune system. That makes us, basically each person's going to have a different ability to fight pathogens. So something that I might be very good at fighting off, you might not be as good at, but there might be another pathogen out there that maybe you're better at, and then I have a little weakness too. And so these different MHCs basically have different ability to bind with antigens, and then some antigens or epitopes of those antigens can basically be better able to attach, more likely a higher affinity. And so because of this, they were looking at, well, maybe some variations within this HLA-A gene made it more likely for people to develop vitiligo, and that's actually where the study concluded. So what they did was a meta-analysis. So basically, there were studies that were all over the board on this. Some had said, you know what, yes, there's definitely a genetic component. Others said, no, it's there's no genetic component to the HLAs and ending up with vitiligo. And so a lot of studies are only done with 10, 20, maybe even 50 people would be a large number of people. So they started looking at the results from databases and compiling the information from trial after trial after trial to try to basically sift through some of this information, figure out what was going on. And they found that definitely if you have HLA-A and you have alleles asterisk 02, asterisk 33, or this AW asterisk 31. So these are the different alleles of HLA-A. And they were associated with an increased risk of vitiligo. So that basically means these particular versions of the HLA-A have a higher affinity for forming self-immune complexes instead of non-self, and so that would more or less give your CD8 cells a better chance of attacking your melanocytes, whereas HLA-A09 and AW19 were associated with a decreased risk of vitiligo. So what that was saying is, all these studies, we found that people that had these two alleles had a much lower chance. Now, there were some other ones that they found that were kind of in the middle of the road, maybe a slight risk or no risk at all. And that kind of goes back to the whole theory that, yeah, we have a whole bunch of different versions of these HLAs that you could possibly get. You're going to get one from mom, one from dad, so you're going to have two different versions of this particular allele. And this is only one of the HLAs that are on your cells. And so some alleles are basically just going to make you more likely to have an autoimmune reaction. And so it kind of brings both theories together, both the genetic aspect and the autoimmune concept. And so, there's got to be a couple things behind this too. You're not going to develop the autoimmune disease necessarily if you have the right genes, but you have the right circumstances and you have the right genes. Now it suddenly comes into focus and you have it. You might have the right genes for it, but and so that means you're more likely to develop that self-immune complex, but the circumstances never come out to where you're displaying that self-antigen and it's not recognized by a CD8 cell, and so therefore you never actually develop the vitiligo. On the other hand, there are other versions of the HLA that can be reactive with these self-antigens. They just maybe have a harder time with it, but the right circumstances. You go through oxidative stress. That's been another thing that was a contributive factor, and that causes the cell to be thrown off. Maybe there was some type of other stressor, like an infection of some sort in that cell, and then suddenly now you have a HLA that's not very, an HLA version of MHC1 that's not very likely to be reactive, self-reactive, and now suddenly it does become self-reactive because of other external circumstances. So this is why it's one of the reasons why it's so difficult to diagnose vitiligo and do research on it, because you're dealing with a trait that's very polygenic. You have multiple factors, and then you also have the immune system basically having to get just the right set of circumstances to have the immune system attack your own cells. And really the immune system's job is to attack pathogens and not harm the host, and this has got to be just this oh so right set of circumstances where now suddenly, hey, we're recognizing ourselves, we're attacking ourselves, and that's a problem. So what we want to see here is, this is their study, more or less the results on which alleles showed a correlation to having a connection to vitiligo. And so if you look over here in this column, which is called the p-value, if you see some of these really low numbers, like for example this gene right here, the AO2, we go across there and the p-value is extremely low, meaning that, yeah, there's a really strong chance that people who have vitiligo have this particular allele. We see this again down here with, this is AW19, I don't remember that being cited, but I know up here the A33 had a really low p-value, therefore that means that yes, it did have a strong correlation. Another one was the AW31 was another one that had a relatively low p-value, and therefore statistically very significant. And we could see here the patients, 8 out of 48 had this AW31, and meaning that, you know, that's a lot of people that have that and then end up having the disease, where only like 5 out of 107 didn't have vitiligo and then had that AW31 allele. So that's a pretty good indicator that yes, if you have that allele, you have a much better chance of developing vitiligo. So this chart, well, these couple charts break down the results they saw from some of these alleles. So if we can zoom in here on this particular one, and you can see this right here is this statistical like average, basically meaning that there's not really any statistical significance. This means you have basically a 50-50 shot of developing vitiligo. But in this HLA-02, we see that the average statistic tells us that, you know, this is definitely above average. It's well over to the side, and that looking at all these different studies, the average tells us that yes, if you have this particular trait, you're going to develop vitiligo. If we look down here, and this one's looking at the HLA-A01, and it's pretty much so right at the same chances of developing vitiligo whether you have this gene or not, or I shouldn't say gene, whether you have this allele or not. And so it's just a slight less. So if anything, maybe it's a little bit decreased, but it's not statistically significant. It's very, very close to just the averages that we would expect to see. So not much of a significance there. And over here we're looking at another one. This one is the HLA-03, and right here again, here's where our expected results are from, like the general population, and we look at the average of all of these studies, and again it's definitely to the right. So essentially, if you have the HLA-A03, you've increased your chances of developing vitiligo according to their meta-analysis. So the last study I'm going to talk about was by D. Rowe and Sina, and that was earlier this year. And what they did was they looked at some blood samples from vitiligo patients and looked at and compared them to control patients, and they did something where they looked at the transcription levels of some genes that are associated with the immune response. They had to kind of pick and choose, but the idea was we're going to look at some of the peripheral blood samples. We're going to go through there. We're going to figure out which particular strands of mRNA are being transcribed more frequently or also significantly less frequently, because sometimes the immune system, when it is turning on, they're going to quit transcribing some parts of the genome to decrease some chemicals, to decrease some signals, and then other parts are going to increase. And so what they did was they measured everything, and they had what they called downregulated or upregulated genes. And so over here, if it's an upregulated gene, because you see more mRNA transcription of those genes, you're going to see a positive number, and you'll see a negative number if it goes down compared to the controls sample. And so here are the different gene symbols that we see. And by looking at these, you could find out if there are some places to treat the disease. This is a really, really good first step in looking at how this disease is attacking, but then ultimately to find a treatment. If you can find some way to interfere with the chemical pathway that's occurring and causing the immune response, and you can block that, then you might be able to halt the progression of the disease, or maybe even completely stop it. The two most of note were the top upregulated genes were two members of the cytochrome P450 superfamily, and that's both right here and the right here. These two, they both had positive numbers, and the reason that's significant is because I have mentioned that there was oxidative stress, and it turns out that these particular enzymes are used in reduction of oxidation within the blood or within the cells. And so, these cells could be under oxidative stress, and that would be why they're producing more of these cytochrome P450s. Top downregulated are multiple genes encoding for interferon-inducing proteins. And so you'll see some of those over here, a lot of these ones with IF in it. Interferon-induced transmembrane protein, and so interferons are going to come along. They're going to react with these proteins, and so the cells in the peripheral blood were producing much less of this for one reason or another. So one thing of note here is this chart basically divides the different processes they looked at. As far as here are the genes that are being transcribed. They can be divided into different biological processes. And so looking at the canonical pathway up here, the number one is an immune response and interferon alpha beta signaling pathway. And that's definitely telling us that there's an autoimmune response. We see high levels of this IFN alpha and beta in this type of autoimmune response. And so remember, it's a logarithmic scale, so 5 tells us that's extremely high. Again, over here, when we look at the GO processes, again, it's an immune system process, and the number one was the number one of those processes was an immune response process. Again, over here, number one was in the process networks. We have inflammation and interferon signaling. Again, pointing towards, hey, there's something going on with the immune system. This is much higher levels of this chemical pathway than what we see in a typical individual. And so the idea was, can we shut some of these down, some of these processes, and find a way of reversing the disease and getting the immune system to stop attacking the melanocytes. In this particular graph, what you're looking at is comparisons of a control. So this is a control group, and here's the vitiligo group, and here are different genes along the sides. And if it's red, that means it's been upregulated, and if it's in blue, it's been downregulated. And so you can see, for the most part, a lot of these genes are being upregulated in the control, meaning that these genes have been turned on. They're doing something in most of these individuals. And we look over here at the vitiligo individuals, and many of the genes have been turned blue. And the bluer it is, the more it's been downregulated. And obviously you have extremes on both ends, but the general pattern is when we look at the controls, things are being upregulated. When we look at the vitiligo patients, things are being downregulated. Same thing over here with these other different types of processes. And so notice they're looking here with the immune system. Some of these processes are being turned on over here in the controls, where they're being turned off in the vitiligo, meaning that basically we're transcribing less. Now, just because you're transcribing less doesn't mean that you're turning off the immune system. It just means you're turning off a signal, and that signal might be something that's saying, you know what, don't attack at this point. And so since there's less of that particular chemical pathway being made, now what happens is the immune system responds to it. So one thing I do keep forgetting to mention is, for example, it has here response to cytokine, and we have 28 DEGs. And so what that's saying is there are 28 different differentially expressed genes that they're looking at, saying that there are 28 genes that, if you look at the control, they're being expressed one way, and you look at the vitiligo patients, they're being expressed totally differently. And it's a response to cytokines, and so that's very significant because, as we all know, cytokines are the way that the cells communicate with each other, particularly in an immune response. That's very important because it's the way that the immune cells know whether to attack or not attack one of our own cells, or whether to attack or not attack a pathogenic cell, or to recognize antigens. And so this is basically one pathway they started looking at in detail. This is interferon gamma, or IFN gamma signaling pathway. It's a top canonical pathway, and it's one of those ones that they looked at in the blood profile and found that it was very important. And so we have a couple of genes that they have circled here in black. And so these are some of those target genes that they want to start looking at. These were the ones that were downregulated and have some connection to the immune response. And so what they would want to do is see if these are some of the targets that they can look at and try to correct the immune response. You're going to see on the next page that they mention a couple of these. For example, one of the ones that they suggest to target is the STAT1. They start looking at some of these other ones as well. Basically, any of these that were circled, they decided these were good targets for trying to correct the problems. But the number one that they mentioned was STAT1, and you can see how it basically has all these spokes going off to these other molecules and other genes that are being transcribed, or they lead to other proteins that are doing something, interacting with other chemicals within the immune response, and so on and so forth. And so they basically found 5 possible molecules to target for therapies. And so these are the different molecules here. And they all connect to processes in the immune response. And if you look at that previous chart, these were all mentioned on that chart specifically. So yeah, that's the end of the presentation. These are the different works cited or articles that I reviewed in creating this presentation. I talked about a few of them in specific, but others were kind of just informative and just contributed to the overall knowledge. In particular, I used this particular article. I did look at this article, the one that showed the coexistence of two autoimmune diseases, quite extensively. It is rare to have both of these autoimmune diseases together at the same time, yet they use a lot of the same chemical pathways. So what are the differences, and how does that lead to the difference in symptoms expressed? But there wasn't as much there to talk about, so I just kind of excluded that. Some of these other ones were just general knowledge. And this last slide was where I got most of my information from. Most of the ones that I was looking at specifically, particularly this one right here, the HLA, that was the genetics one, and then this top one up here. This one connected back to that psoriasis article that I had on the last slide. Interesting, but not as much information there as I wanted to add to the presentation, just kind of peripheral. Thank you, and I hope you enjoyed. Please feel free to comment or give any feedback. Have a good day.